Hello, my name is Volker Thoma, and welcome to my talk, Non-Invasive Brain Stimulation of Right Dorsolateral Prefrontal Cortex Enhances Cognitive Reflection Performance. This work was Daniel Edgecombe's PhD project. Now, I start us off with a math puzzle that you might have come across before. A bat and a ball cost $1.10 in total. The bat costs $1 more than the ball. How many cents does the ball cost? Now, this was one of originally 3 items of Frederick's cognitive reflection test. The intuitive answer that comes to mind for many people is 10 cents. But also after some thinking, usually people are able to say the correct answer, which is 5 cents. Now, this was taken as evidence for dual processes working in the human mind. One process, type 1, is associated with the intuitive wrong answer and given as evidence for heuristic thinking. It's automatic, that's why it's the wrong intuitive answer, because it's also an easy answer that comes to mind easily. Now, type 1 responses can be monitored and inhibited by type 2 responses, which are then associated with the correct response. So this is taken as evidence for slow, effortful thinking that gives you a more reflective response that is usually correct. So we are interested in whether this distinction between these processes can be disentangled. So the monitoring and inhibition of prepotent type 1 responses, and then followed by algorithmic thinking may be linked to working memory performance. In order to do that, we wanted to employ neurostimulation, in this case TDCS, transcranial direct current stimulation, which is a non-invasive safe technique for temporarily inducing a change in neural activity. So you apply electrodes to the scalp, and via a small current you can stimulate then the cortex. Anodal TDCS is linked to increases in neural excitability because it depolarizes neurons' resting membrane potential, and cathodal TDCS decreases neural excitability. We targeted the dorsolateral prefrontal cortex because it is also associated to the processes we are interested in. So the left DLPFC with self-regulation, working memory, and the right DLPFC with inhibition, impulsivity control, cognitive control. In previous TDCS work using judgment decision-making tasks also has targeted the right DLPFC. So our research questions were, does neuromodulation of the frontal brain areas affect reflective thinking performance? And is there a difference between left and right DLPFC? And can we say something more about type 2 processing? So the monitoring inhibition of type 1 responses versus increased working memory performance to mentally simulate certain outcomes that get you the correct answer. Now, we invited people to come to our labs to perform certain tasks on the computer while or before they were, after they were capped with a neuroelectrics cap that had battery-driven electrodes delivering the transcranial stimulation. So in our first experiment, we had 54 participants, and the delivered intensity through the electrodes was 1.5 milliamperes. In group 1, we stimulated the right DLPFC with the anodal electrode over F4 and the return electrode above F3. And in group 2, we stimulated the left DLPFC with the opposite montage. So the anodal electrode above F3 and the return F4. And in the sham group, they were capped, but of course, no stimulation was given other than a very brief ramp on at the beginning and ramp down at the end. So they felt some tingling. The task and measures that we used for measuring reflective thinking were, for once, the incongruent base rate vignettes battery. So here you show people descriptions and ask them to make judgments about certain category referrals. So in the blue example here, you have 1,000 people, 150 architects, 850 are taxi driver. Of these 1,000 people, you randomly sample Stephen, and Stephen is very shy and withdrawn and so forth. And the idea here is that people ignore the base rates and say it's more likely that Stephen is an architect rather than a taxi driver. Then we had a cognitive reflection test battery, the CRT, and you already saw an example of that. So each of these 10 items that we used had an intuitive answer that came to mind quickly, but after some thinking, a more correct answer should be able to be reached. And then finally, we had logic syllogisms. So you would be familiar with this, two premises and a conclusion, and people are asked whether the conclusion is logically valid or not, given the two premises. We also asked people to fill in questionnaires about cognitive characteristics, so individual differences that are known to affect cognitive reflection. For example, the actively open-minded thinking scale measures the willingness of people to consider new information and change their first judgment, remain open-minded. Then the rational experiential inventory, which measures need for cognition, so how much you like solving puzzles, for example, and the National Adult Reading Test as a proxy for IQ. Here you can see the experimental procedure, so 3 phases. Pre-TTCS, people fit in these questionnaires that I just showed you. Then they were capped. The stimulation was given at rest for about 20 minutes. The cap was then removed, and then post-TTCS participants completed the task on the computer. Now, here a descriptive table on the results for the demographics variables and the cognitive characteristics scores. And if you scan quickly, we'll see that there are no difference between groups that is anyway significant. So we can safely say that participants we had in this experiment did not differ across these cognitive characteristics. And to the results, so we have 3 panels here. Panel A on the top left shows on the left DLPFC stimulation, the black bar, and the white bar, the sham condition, and the gray bar, the right DLPFC stimulation group, that there was an advantage, higher scores after right DLPFC stimulation compared to sham and left DLPFC stimulation. In panel B, for the incongruent base rate vignettes, very similar results, an advantage or increased scores in the right DLPFC compared to sham and left DLPFC. And at the bottom, panel C, we have the results for the logic syllogisms. Here we followed Evans calculating a logic index. And we can see here that the left DLPFC stimulation actually had a lower logic index, so less of a syllogism performance compared to sham and right DLPFC. So to summarize experiment 1, we found effects of a nodal stimulation of right DLPFC on reflective thinking performance, so higher performance on the CRT and incongruent base rate vignettes, but no effects of boosting performance from left DLPFC. So in study 2, we wanted to first replicate those results, but we also wanted to see whether it is possible to say more about the processes assumed to underlie type 2 thinking, monitoring and inhibition versus algorithmic thinking, which we measured with working memory updating. We also controlled, in addition, for impulsivity, so we employed the Barrett impulsivity scale, and we had 2 sessions for some of the participants. So this is the design of experiment 2 with 48 participants. Group 1 had 2 stimulation sessions, about 24 hours apart. Group 2 participants came in twice again as well, but in the second session, they were capped, but no stimulation was given. So it was a sham condition in the second session. And the third group came only in once. They were capped, but no stimulation was given. The task, again, the incongruent base rate vignettes. Again, we used the cognitive reflection test, this time with 2 times 10 items, because we had 2 sessions for some of the people. And we also employed the verbal cognitive reflection test, which was similar to the classic cognitive reflection test items, but with no numerical component. And we also employed working memory updating task, the Sternberg test and the NBAG task. Again, here our experimental procedure is similar to experiment 1 with the setup phase, stimulation at rest, and data collection. And briefly, as I said, we had the incongruent base rate tasks 2 times 10 sessions per session. So we had 10 incongruent and 10 neutral items per session. We used the extended CRT, so numerical CRT items like the bat and the ball question with additional items from other sources. And then we used the verbal cognitive reflection test. And in blue, you see an example how it works, very similar to the classic CRT items with a lure and intuitive answer that can be overridden and the correct answer that should be obvious after some thinking. So for the results, again, descriptively, the cognitive characteristics that we measured, and again, we find no differences except a slightly higher need for cognition score in the sham group, but because that should work against our predictions anyway, that should be fine. And here are the results from the first session. So this is a straight replication of experiment 1. In the left panel, you can see that after stimulating the right DLPFC, scores are significantly higher than in the sham group. And in the right panel, you can see the same result pattern for the incongruent base rate vignettes. Again, a boost in the stimulation compared to the sham group, and no difference for the verbal CRT task. Then the next slide shows you the results for the second session groups. So here we compare when they come in a second time, the stimulation session versus the sham group session. And again, we find that the CRT here is high in the second session compared to sham in the second session. And similar result for the incongruent base rate vignettes. Again, a boost in the second session compared to sham in the second session. But again, no effect for verbal CRT scores. And finally, we look at the repetition effects from session 1 to 2 when they came in twice. In the left panel, you see that stimulated first and then second again gives you a boost in the second session, yet again for the cognitive reflection test, but not the incongruent base rate vignettes. And comparing that to group 2, where the second session was only sham and there was no performance change, gives us sort of the confidence that we can see that the increase in the CRT scores in the left panel being stimulated twice cannot be due to just repetition effects, practice effects. Finally, working memory updating results. Here we analyzed the same comparisons as we did for the reflective thinking tasks, and we find no significant effects or trends. So to summarize, we find significant effects of a nodal stimulation. So increased cortical excitability in the right DLPFC boosts reflective thinking performance, specifically for the numerical CRT and the incongruent base rate vignettes, while we control for thinking dispositions and other cognitive characteristics. We also find evidence, at least for the cognitive reflection test, of additive effects of stimulation across 2 sessions. And as we don't find any effects on working memory, updating, performance boost, I think this is tentative evidence for enhanced inhibition of type 1 processes that explains our results for the right DLPFC stimulation effect. Thanks for your attention, and also thanks to our participants.